The overall goal of this procedure is to quickly virally transduce GABAergic interneuron progenitors, MGE cells, before transplantation into a host for in vivo studies. This is accomplished by first isolating MGE cells from a Cre-positive donor embryo. The second step is to quickly and efficiently label isolated MGE cells with a virus engineered for a specific purpose, such as labeling a specific cell type. Next, the transduced cells are transplanted into a host and allowed to develop in vivo. Ultimately, fluorescent microscopy or other techniques can be used to assess the transduced and transplanted cells at the appropriate time.
One of the main advantages of this approach over other techniques like direct injection of virus is that MGE cells migrate a long distance away from the injection site, which is a site of injury, making it possible to study the cells in a more normal environment.
Since MGE cells are progenitors of cortical interneurons, a cell type involved in neuropsychiatric disorders including autism, schizophrenia, and epilepsy, we think that the techniques described here will actually help get at the biological underpinnings of some of these neuropsychiatric diseases. In addition, this technique provides a rapid manner to genetically modify these cells for in vivo studies.
After preparing lentivirus and dissecting the MGE according to the directions in the written protocol, collect both hemispheres of the MGE and put the tissue into two 1.5 milliliter collection tubes containing 500 microliters of DMEM with 10% FBS. Keep the samples on ice.